In this video, we will discuss about biogenesis of microRNAs, or how the microRNAs are formed within the cells. In our previous video, we have discussed how the microRNAs could be one of the therapeutic idea to target the cancer or tumor cells. So there are different ways by which this biogenesis of microRNAs are being characterized, and one of them is canonical, and another type is non-canonical pathways. So in the canonical pathways, what they will do is, and before going to these different pathways, we need to understand these microRNAs can be intragenic. That is, they can be formed within most of the intron or non-coding part of the gene, or some coding part or exon of the gene. However, there are some microRNAs that are formed from intragenic that are intergenic. Previous one is intragenic microRNAs, and this next one is intergenic microRNAs, where the microRNAs are formed independent of the host gene to which they are finally going to target. So let us start with this canonical pathways. So this is the pre-microRNAs that are formed, the transcript that are formed within the nucleus, and after that, what will happen? There is a microprocessor complex that contain DROSHA and DGCR8 is mentioned here, that helps in the maturation within the nucleus, and also helps in the transport of these microRNAs into the cytoplasm, where these pre microRNAs then interact with this dicer to form a complex, that is miRSC complex, that can finally act on the mRNAs to either inhibit. In most of the cases it will inhibit, but in some cases it can also overexpress some kind of mRNAs. So this is the canonical pathways where more maturation is occurred within the nucleus. Now, in the case of non-canonical pathways, it is characterized into two different parts. First one is this shRNA or short-range RNAs. It will also interact with the microprocessor that contains DROSHA and DGCRA. However, and also however, the maturations is not occurs in within the nucleus, and they will be transported from nucleus to the cytoplasm with the help of this exportin 5 and RAN-GTP, and there the pre-microRNAs then interact with this EGO2 for the maturations, and thereafter it will form a complex, that is miRSC complex. In other case, the this other case such as this midron non-canonical pathways, where the microRNAs interact with the spliceosomes, so that only the micro so that the microRNAs contents are being exported out of this exported out with the help of exported out with the in a similar pathway that has been followed by this canonical pathways, and in this case also the maturation is occurred within the nucleus, and after that it will interact with the dicer and then form miRSC. So in the last third non-canonical pathways is about this M7G pre microRNAs. So in this case, microRNAs are been modified to this M7G, and here also the processing is not occurred within the nucleus. However, it will be transported into the cytoplasm with the help of exportin 1 and nucleus, and after that it will interact with the dicer, where the that will help in the further processing. Um, One thing I need to correct is here also the processing will occur within the nucleus, and then it will export out to the cytoplasm, and then it will interact with the dicer to form miRSC complex. And this miRSC complex, that, as I have already told you just a moment ago, that it can interact with the microRNA, it can interact with the mRNAs in most of the cases it will inhibit the mRNA translation. However, in some cases it can lead to overexpression of the mRNAs also. So, and during this process, after this miRSC is and this interactions is this that is translation inhibition is usually happened by affecting this EIF 4F complex formation. It is essential for this mRNA translation, and in some cases this decapping of this miRSC complex will also occur using this decapping complex. This decapping of this miRSC complex will leads to the unstability or will cause instability in this miRSC, and in that case the miRSCs are become unstable and it can be degraded. And the mRNA degradation is shown in this case that this miRSC interact with the transcription factors such as this decapping complex, and that it will help in the mRNA degradations using this XRN1. So, and also so the things that I have mentioned about miRSC instability is actually for this mRNA instability. That is, miRSC interact with this complex to form a decapping complex, and that will degrade, destabilize the mRNA, and that will lead to the mRNA degradations, because of the such destabilization can be occurred by affecting the poly-A tails that are usually at the end of this mRNA, usually found at the end of the mRNAs, which helps in the stabilization. So by degrading that, it will help in this destabilization of the mRNA that will lead to the mRNA degradation. And that has been shown here, that the decoupling complex, this translation initiation will be inhibited by this decapping complex, as it will degrade degrade the stability, that is poly-A tail, either poly-A tails, so that the translation initiation will never be able to begin. In that case, this is the example shown here, at GW128.2A complex, and also the CCR4 not complex helps in this destabilization of this mRNAs. So I hope you got an idea about biogenesis of miRNAs, microRNAs. If you need to understand more about this biogenesis, you can go biogenesis or development of microRNAs. You can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.